So I have a quick presentation and a tutorial to do, but our priority is to answer questions. So if you have any questions, just you can interrupt me, and if you brought your questions, just open your mic or send through the chat, okay? And then we can answer or try to answer. So welcome to the computational support session. This first day, we will talk about genotype calling. And I split this in two sessions. One is the SNP calling, and the other is dosage calling. So this is introductory presentation. And the computational support team is me, Gabriel, and Jikin, and also Maria, but Maria is not here today. She will be in the next sessions. I should send you also our emails. So if you want to send questions to us, you can also. And it's important to remember that we have also the Slack channel that's still there, and you can send the questions through there too. The idea of these sessions is to getting you prepared for the workshops in January and answer questions that maybe you have from the other workshops, and also provide technical support and computational support in installations and how to use the softwares. And most of them will involve like molecular markers and polyploid species, of course, because it's a polyploid workshop. And with these molecular markers, you see that people will discuss many things in the workshop, from linkage mapping to genome selections and maybe gene expressions, and all involving molecular markers. So to do all these studies, we will study like genomics variation and ways to capture these variations. And we have many types of molecular markers. And now in resources, we have been seeing more and more of many markers coming from arrays or sequencing data. And there are differences between them. Arrays, for us here in Roses, we consider this much more expensive for us. But also it's, and I can tell because usually me and Jikin, we evaluate this data, and array data is pretty good. So, and it's easy, straightforward. You don't need, you probably, in my presentation, you could skip to the last part because you will receive like these files with intensities of both alleles, and you just go and do the dosage calling. And last year, Roland made a tutorial, a really good tutorial with FitPoly, their software. And I put a link for this tutorial in the last slide of this presentation. If you want to deal with array data, do the dosage calling in array data, I suggest to see that tutorial. And I think we have also a video of his presentation, and you can follow you can find it in the Polyploid website. And the method that I will focus here, because we will face more technical issues, is dealing with sequencing data. And we have been using that because it's cheaper, and also because if you use arrays and the chip was made for populations that are very different from yours, you are limited to the SNPs that are there. And using sequence data, you can find different polymorphisms. And so this will be the subject of this presentation. And if you want to do experiment with sequencing data, you need to think seems like planning the experiment. You have many types of technologies that you can use, many methods, many protocols, and also later you'll see many softwares that you can use to evaluate this data. And the genotyping by sequence methods come for balance between the amount of individuals that you sequence and the coverage of your sequencing. So instead of you sequence the entire genome for all the samples that can be very expensive, you reduce the representation of the genome with enzymes, like restriction enzymes, and you just sequence part of the genome. So you can have more depth, more coverage for each one of the loci. That for us, for doing genotype calling, is very important. So there are many things that you need to think before you start the experiment, like which if you use a genotyping by sequence or not, what makes sense for your study. And if you use genotyping by sequence, which type of enzyme you use, and how is the capacity of your sequencer, and how many individuals you want to sequence. So many things that you need to put in balance and decide which will be your protocol. So I put here a little overview of the method. So if you're using genotype by sequence, you first cut the genome with restriction enzymes. You put adapters. You use PCR to replicate these sequences. And in this part, we can't help too much because it's not computational. It's more like wet lab job. But in the second part, from the quality control, is all sequences, is data. It's FASTkey files, and then we will start from here. Okay. So once we receive the sequence, we will check if the quality was good and do all the cleaning that it's possible before start the analysis, and then take the sequence, align to a genome, and then perform the SNP calling. You will try to identify the places that there are polymorphisms, and doing that, you will count the number of reads with one of the allele and the other allele. So the reference allele, the one that's it's equal to the reference, and the alternative allele that's different. And once you have this measurement of how many alleles, how many reads you have with which one of the alleles, you can start to identify which individuals are heterozygous or homozygous by each loci. So I will call this genotype calling or dosage calling. And I included here just an example of how would be the alignment of a whole genome sequence. So it will cover all parts of the genome. So each one of the gray lines are reads. And you can see the polymorphisms are the color places, and the colors are the alternative alleles. And in the same place, the ones that keep gray are the reference allele. So for doing the SNP calling, is that okay, we located where the polymorphisms are. And for doing the genotype calling, we will count the number of reads that have one of the alleles, that the alternative allele, and how many is the reference allele. And we will do the proportion and decide if it's homozygous or heterozygous. So this will be whole genome sequence. And for genotyping by sequence or reduced representativeness genome methods, you will see that the sequence comes only in the places that there are the cut site. And this is an example of genotyping by sequence, and this is exome sequence. That's other way to reduce the representation. And when, like now we are talking about polyploids too, no? So when it's a diploid individual, it's defined if it's homozygous or heterozygous. It's easy, like if you have the two alleles there, probably it's heterozygous. And if you have only one of the alleles, are homozygous. But when you start to talk about polyploids, it's not only one class of heterozygous. You can have other types of heterozygous. You can have like only one of the reference alleles, and like, for example, these ones here, only one of the reference alleles and all the others as an alternative. Or you can have two reference and two alternatives, or the opposite, like one alternative and two and three reference, or all references. This is for tetraploids. So you have five classes of genotypes and 3 possible heterozygous. In an hexaploid, you have even more. You have like 7 possible genotypes and 5 possible heterozygous classes. Okay, so we need not only to distinguish which ones are heterozygous and which ones are homozygous, but also the dosage and the heterozygous. And this is a very classical graphic that you use in dosage calling. Like we put the counts of the reference allele in one of the axes, and the other axis we put the alternative allele. And each one of these dots are individuals. So the individuals that have only reference allele will be in the x-axis. The other that have only alternative allele will be in the y-axis, and the ones in the middle will be the heterozygous. So this will be for diploids. For tetraploids, we'll have more axes, and we can split the classes of heterozygous like this. And in hexaploid, we have more axes. So you can see here that this is theory yet. And when you increase the number of possible classes, heterozygous classes, harder is to distinguish between each one of the heterozygous. And this is the theory. If you go to real data, this can be even more complicated. This is a real data for diploid. So you need to try to fit the, like which dots here are heterozygous and which dots here are homozygous. And this is also for a diploid, but with higher def. And check that if you have, like, if the dots are far from the axis, it's easy to distinguish each one of them. That's why, like, higher def sequence, it's usually better. And this is a tetraploid scenario, and you have more axes. So the goal of the dosage calling software will be do this job for you, like, and then, like, we have many of them, but each one of them will do this in different ways. So we will discuss two types of software here. One is the dosage calling, and they are, I would say, you will face less technical issues running them because most of them are in R, so it's not difficult to install, and also doesn't require much memory and computational resources to run. So I included only Updog in the tutorial that I will show you, but Supermassa and FitPoly and PolyRed, it's not difficult also. And Supermassa, we talked about this last year too, but there is like the software is much harder to install because it was not updated since the last time that was released. So it's still in Python 2, and this can be complicated. And I would say, like, go ahead and do the PolyRed, Updog, and FitPoly, that the results will not be worse than Supermassa. And so we will discuss the dosage calling and also the SNP calling. That's this part of identify the loss set and count the number of reads of each one of the alleles. Okay, so this is the theory. Now we go to the technical issues. So dealing with sequencing data, you will have large files. Different from the dosage calling software, you will not receive just the markers or the number of each one of the alleles. You receive like FASTQ files that can have gigabytes or terabytes, or it will be huge. And this will be difficult to you to do only in your computer and in the personal computer. And also for doing some of this step, like the quality control, the alignment, and SNP calling, you have many possible softwares to use, and they are not in R. Like they have different languages, and they usually run better in Linux than Windows, and they are pretty hard to install sometimes. And also they have constantly updates, and you need to like be aware to always update the softwares if you are using. And so this can be hard even for you to start to get all the software installed. And if you want to try more than one, it's even harder. So the idea here is just to show you an easy solution for all of this. And like for dealing with large files, you will need to use high performance computing or use the cloud like Google or Amazon. And this high performance computer usually have like management system. There's Learn or the SG that you need to learn how to do, because it's the computer will need like put a line to everybody be able to use, and not, you don't break other people process and things like that. It's and it's not so difficult to learn. And And also to solve the issue of you having many softwares, and if you don't want to install these softwares, and if you want to be able to control the versions of the softwares, well, a good solution is using containers like Docker containers or Singularity containers. And there are like repositories available that have basically all the softwares that you need, like all softwares that are listed here. There's a container image available online. So instead of installing the software, you just need to download the image, and then it's done. Use the image. I need to say also that if you are using high performance computing, maybe some of the softwares is already available, because like at least here in Texas A&M, the team that work in our high performance computers, they are pretty good, and they install many of the softwares that we need. But this way you can't control too much the versions, and the recent developed software also, it's hard to install, so you need to be bothering them all the time. So I personally don't like to bother them too much, and then I prefer to use the containers because they provide the Singularity, so it's the only thing that I need. And the difference between the Docker and the Singularity is that Docker will need like super user sudo commands, sudo permissions, and the Singularity doesn't need it. So usually HP systems have Singularity available. And if you use containers, it's good to use like image from known sources. Like the biocontainers are a collection of containers that it's trustful. So you can change all of this by Docker and Singularity. And other issue that you will face dealing with sequence data is that you will need to do many steps. And you here, as I show you, like you need to do the quality control, you need to do the alignment and the SNP calling, the dosage calling, and you need to know how to manipulate the intermediary files between these steps. And this can be a little hard. Like you need to know at least Bash or some other programming language to know how to do that. And an easy way to avoid that, or at least have a resource how to check how to do that, is workflow systems. So probably there are people that are doing the same thing as you do, and they provide the whole pipeline for you to follow. And for the easier way of sharing this and make this reproducible, it's the workflows. So there are many workflow systems available, and they are still developing. One of them pretty famous is Galaxy, and we have others like Nextflow, SnakeMake, CWL, and DeepWL. And you can find these workflows for many type of analysis in these two repositories, the most famous repositories, the DocStore and the Workflow Hub. And the good thing about the workflows too is that you can run it in platforms in cloud, and they also have support for running in HPC. So you have like the Galaxy platform, and the WDL, that's the one that I will focus now, it can be running Terra. Okay, so you can change, I did animation here too, like you can change all this by using one of these workflows. And the other thing is that you will need more computational resources. And if you're using the workflows, you can manipulate how you use the resources better. Like instead of using only your personal computer that has probably the same configurations as mine, that is 4 GB of RAM and 8 cores, you can use the high performance computing it's much more powerful. But by using this, you need to know how to use all the resources. Because like if you're using just the RStudio, you use only one core and you cannot distribute. Like the function needs to be prepared to parallelize the procedure. And usually what workflows, the workflow do that for you. Like we will split your analysis not only in cores, but also between nodes. And this will optimize your time and the resources available. And this is an example, and it's in the tutorial. It was a workflow that me and other collaborators we developed. And the goal of this workflow was to build linkage map. And for that, we go through the sequence data until the linkage map. And so the thing is, like, if you don't want to use the entire system, like if you don't want to use the workflow system and not want to learn any of this, the workflow is also still good for you to check which ones are the best steps for reaching your goal. Like as I am one of the developers of OneMap, so I figured out that was not enough to provide the user only the last software of the sequence, and I also needed to provide the best way of getting the markers for my software. So as a developer, I made the workflow as a suggestion of best practices to get the markers for the software. So if you don't want to use the workflows, you will have at least idea of best combinations of software to reach the goal. And that's the idea. And the workflows are working in layers. So you have the big workflow that it's very complex because we usually put everything that we need inside, but it's made by sub workflows and by tasks. So you don't need to run all the workflow. You can run just the sub workflows and it will work anyway. So in the tutorial, we will just run the three first steps, like the alignment and the snip calling. And you can, of course, run this in TerraView. That's the cloud environment, and probably it's the easiest way. But in universities, we have the high performance computing available for free. So I usually use it instead. And I can run it using the management systems like the Crowell or the Mini WDL or the XWDL. I personally use more the Crowell. And you can run the workflow just with a single line. Just putting the Crowell, that's the software, and the input files and the workflow file. And I included here the links for some of the tutorials. I think I mentioned the Ruland fit poly tutorial at the beginning. It's here. I also have like the Updog and Polyploids tutorials here. And Lindsay Clark also made available a tutorial for doing the snip calling with Tassel and adapted to Polyploids. That's also very nice. And this is the tutorial that I will follow here if you don't have questions. So if you have any questions, I think I will stop right now and hear the questions. I don't know if you have something to chat.
Hi, Chris. Thanks for the tutorial. I'm very new to I run some softwares, but I'm not familiar with Polyrad or all of these. Do you think it's easy someone who is not used to using a lot of computing systems?
Yes, I need to say, like, doing the analysis with sequence data, it's a little bit more tricky because you will need to learn a little bit about Linux and how to manipulate the resources. But for doing just the dosage calling, it's R. Mm-hmm. So it's important to have a little bit of acknowledgement about R, and okay, you'll be fine.
Before the PAC conference, do I need to go through these tutorials and have the software installed in my computer? What do I need to get ready for the conference workshop?
Yeah, you of course you don't need to, but if you want to follow the tutorials, because probably the speakers will talk about novelties in their software, and that I cannot predict what they will talk about. But if you want to run with them, it's better if you have it installed in your computer. And the R package, it's pretty easy to do. It's just install package and it's there. And probably if you want to follow what they are doing, it's better you to wait a little bit, like install the versions right now, but update just before the meeting.
Thank you so much.
Welcome. I have a question in the chat if all softwares works in Windows system or if you need Linux. Again, like the package that are in R, you can run in Windows, it would be not an issue. But if you want to deal with the sequence data, you can also use Windows with some softwares that require Linux. If you use WSL, like my personal computer have Windows and I use WSL for everything that I need to do with Linux, and it's great. But if you deal with sequencing data, you will probably need a high performance computer, and this will be Linux for sure. Okay, so I do not suggest you to run anything of sequencing data in your personal computer. Like this tutorial that I show you, it's possible to run a personal computer, but like with your data set, use a high performance computer. Or the cloud environment also works. I'm not sure about like the other, sorry, I will read the question because I will be answered to myself. But someone's asking if I have suggestions for software package for allopolyploids and autopolyploids. I think it depends of your goal, like what you want to do with this data, because like in tools for polyploids, they show tools for linkage mapping, for key tail mapping, for genome selection. So it depends. I think like for allopolyploids, some of the tools for diploids also work. And for autopolyploids, you will need to use one of these other software. So if you were asking about linkage mapping, like they talk a lot about MapPoly and PolyMapR, and keytail poly is keytail poly. We can list the softwares here, maybe if someone can help me and put all the softwares here that we talk about in the workshop. Other question is there a list of programs package that will be needed for the workshop? We will most be in R. I believe that yes. Most of them will be in R. I think the only person that's not in R, maybe it's me. And I think there is also a software in Python that's the MC-Hap, the haplotyping. If you want to follow my talk about reads2map and try it, and I will be really glad if you do it, you will need Docker or HPRC with Singularity. There's another question that's, can you set different polyploids in the same dosage calling? I work with a crop that goes from diploid to dodecaploid. They're like, if you use like supermassa, and I think PolyRed is also available for doing that, it will measure the dosage by marker. So it will use the marker position and consider the entire population to estimate the dosage for that marker. So if you have a neuploidy, like some regions of the genome with diploid and other regions with dodecaploid, it will be able to capture that. But if you have like many different types of individuals, I'm not sure. I don't know if someone knows if PolyRed's able to do that.
Most of these software, you have to set the ploidy beforehand. So you're going to have to tell it either tetraploid, diploid, whatever, and it'll run the whole data set as if it were in that ploidy. So you kind of have to know that beforehand.
Yeah, so supermassa can do this changes by position. So you don't need to have only one, but the entire individual need to have the same, the entire population needs to have the same ploidy among the loci. And I'm not sure about updates of PolyRed, because I heard that Lindsay was working about estimating different ploidies also, but she will probably talk about this in the workshop if the updates is already.
Oh, he says by sample. So say for instance, if you know like your 100 samples here are all diploid, then you can call them as diploid, but they have to be as like one run. Unless like what Chris was saying, Supermassa is able to per marker estimate ploidy.
Yeah, but me and GQ, we try to do the opposite, like try to estimate the ploidy by individual, and it doesn't work with Supermassa and neither UpDog. So we need other approach. And
yeah, we have similar like issues here in rows too, because we have diploids, triploids, and tetraploids, and we have to know beforehand what they are and then group them that way.
Yeah, I remember that Supermassa, we used to work with sugarcane with Supermassa, and there's no like one individual with a single ploid and another individual with other single ploid. We have many different ploids in the same individual, so the ploid was estimated by loci and not by individual, and Supermassa was developed for that. But to estimate ploid of a single individual doesn't work. Are there all input files for the tutorials on the website, or will be circulated later on? I could not find the input files for the FitPoly tutorial. Thank you. Oh, I need to check. I don't know if the FitPoly tutorial inputs are still in the website. I can check with Delaney later. Sorry, I didn't check it before. Have you quantitatively investigated sequence depth needed to determine dosage with a given reliability? Do you use segregation pattern to call? So for this first question, I will repeat. Have you quantitatively investigated sequence depth needed to determine dosage with a given reliability? We have been using a tool from UpDog that called Oracle. So it can tell you the good depth for according with your data. I think it's a pretty good tool, and I can put the name here. And we have been using for that because I think the depth will depend on many things, like not only the ploidy, but also the quality of your sequence and your samples. Because I can see here from the GPS data that depend of the quality of the DNA that's used in the beginning of the protocol, I will need more or less depth because we'll have more or less bias and other issues in the data. So I like to use this tool, the Oracle tool. And the other question was, do you use segregation pattern to call parental dosage? So these softwares, the UpDog, the Supermassa, the FitPoly, and the PolyRed, they all use the population structure to call the individual's dosage. And you can choose the model that you want. Like I mostly use work with mapping population, so usually it's F1 model. So the software considered this type of segregation to estimate not only the progen, but also change the parents if it's required, or sometimes impute the parents also.
Can I ask a question?
Yeah, sure.
So I missed the first part since I was in another meeting. As you may know, there's a platform called Breeding Insight. So I see this tutorial is a Leo calling, and that outcome from Breeding Insight, we already have the Leo call. Then in order to use Polybreed R, the final version, so we don't need to use the PolyRed for Leo calling if we get marker data from Breeding Insight.
Yes, if you're working with polyploids and like your genotype calling, it's already in dosage, like you can distinguish all the heterozygotes classes, you don't need it unless you want to test other methods, because what we usually do is that we go until the end of our analysis and we check if it makes sense, and we go back and test other methods of genotype calling to check if we can improve our results according with the methods. We keep comparing them. But if you already have all the heterozygous classes, you are ready to go, I would advise you to go until the end of your pipeline and check if is everything okay. Is everything okay? It's good.
Has there any study to show use polyride to make a Leo call dosage call compare with the DART technology, or any pipeline made that call? Since the breeding inside is relatively new, also, is there any study to compare using polyride and the call from DART technology? Any difference, or which one's better?
Oh, I don't know about DART tag. I didn't see any. I don't know if other guys here heard about any study compare them with DART tag. I'm working in these workflows are also for comparing, but it's between softwares and not the technologies. So I'm comparing between PolyRed, UpDog, Supermass, and FitPoly. So how they do, the methods are applying the same data. But if you want to compare like the PolyRed with the DART tag, I'm not sure.
Okay. So last night I watched the 6 minutes or 7 minutes video tutorial, and I'm trying to find some testing data. I installed the PolyRide R package and trying to see whether tutorial I can follow up to prepare for this meeting. I go to your GitHub page, and I didn't, well, there may be that already, but I didn't find that data that I can use to run through the PolyRide. Have you posted any raw data in the GitHub, and then I can run through those?
Yes, I did this tutorial here, and
Okay.
it's also this tutorial it's for using the workflows, but I provide small data sets just to try each one of the softwares. Here I just use the UpDog, but you can use the same VCF for getting the PolyRed calls. I think it's just a few markers of chromosome 10 of our data, Rose data, and 222 samples. So it's enough for using PolyRed, UpDog, and FitPoly if you want.
Okay. So can you please share the link?
Yes.
You may already shared, but I was late 20 minutes.
Okay. Here. Yes, I was planning to go through this tutorial, but if you wanted to keep doing questions, I think questions are priority. And the tutorial is pretty self-explained, so you can do this by yourselves later. And let me know if you have questions. I'm not too familiar with DART tag. I never work with that. I don't know if any of my colleagues work with that, and I would love to work actually to explore, but currently I didn't.
Yeah, that is a technology support. The Cornell University leading the efforts to developing genotyping platform for. At the beginning, it had 6 species, but right now it's more species. They all use the same genotyping platform developed by DART technology.
So do you receive sequencing data, or you receive the genotypes with the counts already?
The raw data is the sequencing data.
So you need to do a SNP calling and a dosage calling.
The raw data from DART company is a sequencing data, but the breeding inside provide us with a allele call already. So that platform developed by Cornell University team. Yeah.
Do you know in the background, does breeding insights, do they use GATK to call the dosage, or what?
Oh man, yeah, that's the quick question I would like to know is the Leo dosage and Leo frequency, how they make a call. And they refer to the DART technology website. And I didn't put that in my mind. I can explain clearly to you, but I feel that would be interesting using the PolyRide to make a call, compare the results with the one from Cornell University team.
Yeah.
So just to make sure, they already provide you with the allele counts for each marker, right?
Yes.
Okay, so I think you can get this information and just plug into any of these softwares, right, Chris?
Yes, if you have like how many alternative alleles and how many reference alleles you have for each loci, this is the input information for polyred, updog, and all the others.
Yeah, that is, yes, they will give us like how many they sequence that, how many allele call for this one, for that one. Yes. Yeah.
Yes. No, thank you. It's my first time working with this sort of data, so I wanted to just ask if anyone else had done that before, and it's very nice to listen on this presentation.
I think I will show you the output file of the SNP calling and genotype calling. It's a format that's pretty famous, it's VCF file, and then we can make it see what about everything that we are talking about. I have an example here in the tutorial. So so in this tutorial, I go through the SNP calling and the dosage calling steps, and this is the result of GATK SNP calling. And it can be very messy, like in the first view, like because it's lots of information. I will, instead of open here, I will open here. I should have this here somewhere. I don't know if I have a VCF here. No, I don't have, but I can show you. Here, I have a VCF here. I have VCF everywhere. Here. So this is just the header of, don't be scared, this is just the header of the VCF, and it contains the description, all information that's contained in the VCF. For example, here it's saying that the AD field will describe the allele depth for reference and the alternative alleles, and the DP will describe the approximate read depth, and the GKey, the genotype quality, the GT, the genotype by itself, the DP. So you have the description of all of them. And depending on the software that you use for doing the SNP calling, you have different number of fields and different information that you can use after to filter or whatever. And so and they also record all the steps that were done for doing the SNP calling. So you can also know, like, okay, this VCF comes from JTK because the commands here. And so if you go and the reference genome information, everything, and here it starts the information. So you have chromosome information, you have position information, you have the ID of each one of the markers, you have which base it's the reference and which base is the alternative. You have, like, if you have like quality measurements, it will be here. If you use filters, you be described if this marker passed it or not. This is the info informations, like describe it up there. So you have many, like JTK likes to put all the information. So you have a big line with all the informations for these specific markers. And the format will describe which will be the next fields. So the first, the GT, is the genotype. The AD, as we saw before, is the counts for each one of the alleles, the reference and the alternative. The DP is the total depth, will be the sum of the AD. The GKey, a quality measurement, and the PL, the probability of each one of the genotypes. So this is a diploid, and I can tell because it's just one bar. It's just one bar. So for example, this is homozygous for the alternative. Zeros represent the reference and 1 represents the alternative. And this has 2 alternatives. And this other here is heterozygous because it has a 0 and a 1 and so on. And this is the count of the depth. So it has 3 reads with the reference and 5 reads with the alternative, and in total 8. And this is the quality in Phred score and the probabilities. And you will see that you have this bar sometimes like this, that's the genotype's not phased. And sometimes you have the bar, the pipe, that means that it's phased. So if it's not phased, the higher number will be always on your right. And if it's phased, it can switch, like it can be not only 0 and 1, but also 1 and 0, because you can distinguish in which haplotype the allele is. So this is diploid. And the example that I put here is a polyploid. It's a tetraploid. So instead of seeing just 2, you will see 4, because it's the measurement for each one of the doses. So for example, this is a homozygous because have like 0 of the reference allele and 7 of the alternative. So it's all alternative. And let me see if I can find here. This is a single dose because it has 3 of the reference and 1 of the allele, one of the alternative, because have 38 of the reference and 3 of the alternative. And so you have many, like this is a standard format. You will probably see VCF files in your life, but you can also have like HapMap file that will have only the counts for each one of the alleles. I think the format that he was mentioned before probably has this information here, the amount of reads for each one of the alleles. And this is the input file for PolyRed, for FitPoly, for Supermassen, and then they will provide you this. According with the counts, they will provide you the dosage.
No, thank you very much.
Welcome. And if we're dealing with sequencing data, GATK Freebase will also do the dosage calling for you. So it's a possibility of using that too. But if you want to use, but they don't use the segregation, the segregation in the population to estimate this data. So it's only based in the counts. If you use PolyRed's, UpDog or FitPoly, they will use the population segregation to help you to estimate it. So sometimes it can help. And I have maybe, I will not run the entire tutorial. I will do just the dosage calling because it's the part that we have more questions with. I have here ready, and I can show you how to do that. So I have this VCF file. That's the VCF file that maybe you guys can use as example for the other ones. And I use, so for installing PolyRed, UpDog, and FitPoly, you just install package and it's done. Easy. And I also like to use this package here that's for the VCF manipulation. So, and I will show you how nice it is. So I just call it library, and then I read the VCF file. So it will tell me how many samples and how many variants I have. This is a subset, okay? It's not the complete data because complete data take time, and I cannot make available through GitHub. And so I want to have the a matrix with the counts of the reads for each one of the loci, total counts, the DP in the VCF format that I just showed you. So if I use this function here, extract GT, I have a matrix with the counts, the total counts for each one of the individuals in the columns and markers in the lines. So for example, this first individual have 40 reads for in this marker position. And I can also have the AD format of the VCF file. And then if you see here, you have the counts for the reference and the counts for the alternative. See for each one of marker individual. And I can extract just the reference or just the alternative using R. So I just split the matrix by the comma. So I will have a huge list with all the genotypes, and I will select the reference. and then I build the matrix again. So I have just the counts for the reference allele for each one of the genotypes. And if you see here, this is the reference matrix, and this is the AD matrix. So I just extracted the reference here. And this is the input for updog. So updog will do the dosage calling, and it requires this reference matrix, the counts of the reference alleles, and the counts of and the total counts, both metrics that are already extracted. And if I try to run, you will see an error here because it says that's supposed to be numeric. And I you know, when I was preparing this tutorial, I saw that this error were here, but I decided to keep it just to you to understand that errors happen all the time. And because like sometimes we build tutorials and it runs perfectly, and people that runs the tutorial later think that's easy, like it was, and then when they try to do by themselves and they find issues, they think that they are doing something really wrong. But no, it happens all the time. Like we face issues, but we just pay attention to the error message and we fix it. And I just made as numeric, and I run again, and then I had another issue saying that my reference, my matrix doesn't have the names, and I need to put the names. So I put the names, and then it's finally running. And I can show you the result. I think it will take some time to run.
Do you have any experience with populations that throw a lot of aneuploids?
Yes. Yes, we have. Roses has this issue.
And you force the euploid assumptions on those populations, and then you get problems, right?
Yes, and it's like these softwares, because they are not ready to deal with that. We can see the issues of it in the linkage map further in our pipeline. Like usually we assume that they do not have aneuploidy, and then we have issues with that later. What we are trying to do is eliminate this data. So detect that, okay, aneuploid is here. So make these genotypes as missing data so we don't have issues in the key-tail mapping further. And until we don't figure out a way to do that. Because like for us, even if we can fix it in the dosage calling, the linkage map software is also not ready to deal with aneuploidy, and also not the key-tail mapping or the genome selection. So we are trying just to identify where they are and try to make us missing data. Oh, it's finished here. So once it's finished, you can see that plots that I show in the theoretical presentation with the axis. So I told them here that my ploid is 4, and it makes the dosage call, the genotype calling according with that axis. And it's another type of codification. The 0 would be the all reference, and 1 would be like if you were going to do as VCF, the 1 would be like this, and the 2 would be like this, the 3 would be like this, and the 4 would be like this. And then you can get all the dosage in a matrix using these 2 commands. And this is the updog approach. And Polyred, I included the tutorial, but you should have the same type of results, like the dosage for each one of the individuals. I think someone's asking if I use JTK for the SNP calling, and if this data are from GBS. Yes, this data is from GBS, and I use many software because like my goal is to compare many software. So I use the workflows to do the SNP calling with JTK, with Freebase, and with genotype calling with Polyred, Supermassa, FitPoly, and Updog. And I'm still adapting the workflows for doing this automatically, all these comparisons with polyploids. But I did with diploids, and we have a preprint about this, like comparing all the softwares with linkage mapping. and for diploids, and we intend to do the same thing with polyploids now. I'm including the map poly in the workflows.
Chris, can I briefly interrupt you?
Yes, sure.
So my question is more so related to upstream of this. When you're in the process of doing SNP calling, do you do any sort of estimation of ploidy to remove aberrant individuals that might be aneuploids or diploids as a result from some sort of missegregation or aberrant meiotic process? Are you doing any sort of diagnosis of the ploidy level prior to doing the dosage calling?
Not yet. We are thinking about ways to do that yet with GBS data. And so far, what we do is testing segregation before going to other steps. But we can only do that because we work with mapping population. If we would work with germplasm, I'm not sure how I would do that, but it's the only control that we are doing by now. But we are exploring others.
Yes, I'm more wondering what sort of quality control metrics are being implemented prior to making assumptions about the kind of the structure of the population that you're working with. In this case, you know, if you're just working with biparentals, you might want to diagnose whether you have contamination and how you might go about doing that.
Yeah, about contamination is easier than aneuploidies because like we can solve this with a simple PCA. Like, and I think some of the softwares have, I think Polymap R has this function to just do a PCA and see if you have contaminations and if all the clouds of the progen it's between the parents, so you can eliminate contaminants like this.
Do you think PCA, if the input matrix is a matrix of SNPs by individuals, what if certain patterns emerge from a PCA that may not necessarily be related to contamination, but is it possible, do you think, that patterns that might appear in a PCA could be a result of some sort of genomic feature of that population?
I'm not sure. Do you know something about that, Chicken? Did you see something weird in your PCAs?
No, we only really use PCAs to pick out the what's that called? All the individuals that don't belong. So kind of this wasn't using sequencing data. This was using array data. We were trying to do two mapping populations that were interconnected, but then like as a very first step, we just did a PCA and we saw that there were a bunch of selves. There was an off-target population that didn't belong. There were individuals that didn't belong to any of them. And what I saw, because as an interconnected population, was like a triangle. So there were the parents were the vertices of the triangle. The two populations were in the middle, and then what was all the off types were like grouped into clouds separately from that, or they grouped with the parents if they were selves. So that's what we use it for. But as for your question of like, before we go into UpDog using sequencing data, I don't think we don't have a method of like looking for aneuploid yet, like before you do the dosage calling.
Right. I guess I'm wondering if PCA would be the only way, especially if you have, say, two potentially highly related parents in a species that has, say, some sort of aberrant or structural genomic features, right, that may be restricted to one parent or another, if there would be other approaches to identify potential contaminants, and if PCA would be...
What we can see clearly when there are aberrant things, like can be aneuploidy, can be that data, it's in the mapping. Like when we build the linkage map and build the haplotypes, there are haplotypes built that's perfectly like it's as expected. We just see one single break in the progeny haplotypes, or maximum 2 as expected. But there are some individuals that we can see many breaks. And so for us would be many genotyping errors, but also can be aneuploidy. So what we do, what we are trying to do now is like build the linkage map until the end, get these markers, these positions, and then go back to the raw data and make them as missing. So they at least don't disturb our linkage mapping, because linkage map is a great tool for identifying what is wrong with your SNP and genotype calling. And so if you can do this upstream, we can do at the end, and then we go back and fix what's wrong.
All right, thank you both. I won't take up any more time, but I'll shoot you both an email later.
Can you use the total read counts from different loci, or perhaps a mean read count for a given locus over the population, well, over the individual, sorry, to estimate the likelihood that a given marker is aneuploid or you know, an aneuploid dosage because of translocation duplicate deficiency?
I don't know.
Well, I'm going to hop in. This is Oscar, and good to hear you, Charles.
Yeah, this is Charles.
And I guess good cytogenetics question there. We think that we might be able to detect some of these chromosomal anomalies and copy number variations from the data. We're on the process of trying to figure out exactly how to approach it. And again, it depends on the quality of the data and the types of data that you look at, whether they are sequence counts or luminosity differences from array data, right? So we're thinking that we need to look at the whole chromosome or pieces of chromosomes to get a feel for that, rather than individual markers per se, right? So we're exploring that, we're testing it, and as soon as we get something worked out, I guess we're gonna try to get that out, okay?
Okay, I was just wondering how much time I need to invest in a simulation study, but anyway, yeah, I have my Zephyranthes in mind, and of course they're translocation heterozygous, and they're octoploids and decaploids, and they're apomectic, and all these other problems. But anyway, I have a tactic to deal with them.
And as I said before, we are looking into try and maybe get a method that gets us down to smaller pieces or segments that we think will be reliable to look for duplicate deficiency situations, for example. Yes, that would be the next step. Right now, we're thinking about starting with ploidy, aneuploidy, and then going down the line to smaller areas of the chromosome. Yeah.
Okay, thank you.
I have other questions here in the chat. And one person asked is your general pipeline through SNP call as deployed in GTK for base and dosage call in updog polyred, etc. Or do you set ploidy as the SNP call in stage 2? So my pipeline, so far you can do the SNP and the dosage calling with polyploids and diploids, but I'm still working on that. So until January would be also possible to do linkage mapping with MapPoly and in the pipeline. So the idea of my the readstream map is to measurement the quality of upstream tools by the linkage mapping. So as I was saying to Josh, sometimes we just know if the SNP and the dosage calling was good once we see the linkage mapping. So I wrote the workflows to do everything automatically until the end, so we can pick the pipeline that we will choose by the quality of the linkage mapping. And I did this for diploids, and our preprint is about that for diploids. And now we were doing the same thing for polyploids too.
Hi, Chris. I have one question. So in this VCF file, you have actually a diploid genotype. So in your genotype matrix, you have allele depth for 1 and alternate allele. But what you're showing me from multi-dog function is that you can actually use that information and and actually tell what the dosage is? How does that work?
Yeah, so it's the information that I go in. It's not actually diploid, it's just biallelic. It's just the information of how many counts of the reads are for the reference and how many counts are for the alternative. And this will create this type of distribution here, like showing in the graphic. So I will put the individuals that have only reference counts on the x-axis and the alternative in the y-axis, and the ones that have just a proportion of them will be in between, and there will be different classes of heterozygous. So how UpDog works is the ones that's closer to each one of the axes, they will categorize as one dosage. So the 0 will be only like it's a tetraploid individual and will have only reference alleles. And the 1 means that one of the homologs have alternative, but the other 3 has reference, and so on. So these have 2 homologs with alternative and 2 with reference, and so on, according with the proportion of the counts. So I think it was here's the image, but I want to show you that. So I'm just counting the number of reads that align in the same position. So for example, this would be homozygous for the alternative because it has 1, 2, 3, 4, 5, 6, 7, 8 counts of the alternative in this position. So this individual in this position is homozygous for the alternative. If this position here, you can see that they have some reads with the reference and some reads with the alternative, you can see. And this is probably heterozygous, but which type of heterozygous, if this is a tetraploid individual, will depend on the proportion of alternative through reference. Gotcha?
Okay, yeah.
So you're just looking at the ratio of the allele counts, and that's why it's so important in polyploids to get a deep enough coverage so that you can get the ratios.
Yeah, don't aim for 4x coverage.
Yeah.
Thanks.
I have a question.
Sorry,
okay, I think now. How do you know if you have enough coverage for this?
So I mentioned a tool in UpDog that calls Oracle, that you can go in with your matrix of counts of alternative and reference, and they will say the probability of your calls being right. And it's a good tool. But like in our experience for our tetraploid data, depth of 50, it's already good. We could build good maps with like just 50 read, like in mean of 50 of coverage. But use the tool, is a good tool.
Thanks.
And it's ploidy dependent. So the higher your ploidy goes up, you're going to need higher depth to be able to tell the classes apart from each other.
And it's a nonlinear increase.
Shankar again here. So while generating this BCF file through GATK, you probably specified either ploidy equals 2 or didn't even specify ploidy, I don't know. But if I run the GATK with number of haplotypes equals 4, something like that, I would expect to get very similar metrics that I'm getting from multi-doc, because when I specify ploidy of 4, it would probably give me 4 alleles through GATK. Am I getting that correct?
Yes.
I know there might be some differences in what doses we're getting, but that's what you also mentioned, right? GATK could actually be, if you know if it's a triploid or tetraploid or pentaploid, you can specify that beforehand in GATK and actually get some sort of dosage information from there by looking into the genotype section.
Yes. if you in the tutorial that I was let me just come back here. I included here how to run the GATK. So you can specify here, you see the ploidy of your individual, and it will provide to you the dosage that you can use. And it's the same thing that will be provided by the dosage calling. And GATK only use the data information. And if you use UpDog PolyRed, probably some individuals will be different because they use the segregation of the population also. And this can kind of improve or not your data. So the idea is to compare them. As I showed here, like this is just another type of codification for the same thing. And in the GATK, you will see something like this in the genotypes. And this will be the same thing as 0 here, just the reference. If you have something like this, which one? Here, it's a single dose. So it's the same thing as the number 1 here. And it provides the same information. Okay, the minimal depth for the tetraploid dosage, again, like for us, 50 worked for one data set, but it depends of the quality of your data. So I would suggest you to use the Oracle to check your probabilities, okay? Because it's a good tool and it's better than just say a number here. For us, 50 worked, but I don't know. And if you increase the ploidy also, as other people said here, it will require more depth. I'm not sure if do you know, Gabriel, if other package like PolyRed can deal with non and magic population?
I'm not sure about this.
Yes, just sorry about that. I didn't check the tutorial about that. But if they do, you can see this easily in the models that they allow there. And maybe this too can be in the help function, like multi-dog. They describe here the type of models that they can receive, and PolyRed also have the same thing. So just check their manual, okay? Is there any tool to compare genotype calling done using different rows on the same panel? Oh yeah, use the workflow if you're doing with sequence data. Does there has any software package could consider dosage effect during genomic selection analysis? Yes, I think StageWise does that. No, Gabriel, Jiken?
I don't know, Jiken, do you know that?
Not familiar enough with StageWise yet.
Mm-hmm.
StageWise does have like additive versus dominance effects. So the effects of multiple of the same allele, but I'm still playing around with that software.
Yeah, I'm not sure if I got the question correctly, but if you were meaning considering deployed level of on the dosage information for genomic selection analysis, this can be done when you use any matrix with dosages. So if you proceed with a GBLUP model with dosages, you get the dosage effect for each marker. If you run an RBLUP or a GBLUP, you can translate from one to the other. I think that Jeff Endeman, which is the developer of StageWise, he developed another package which does that for dosages. The package is called RBLUP. And I think you can use dosages, but as soon as you adjust any GBLUP model using SRAMO or SOMER package, you get the same dosage effect. So that's possible.
I think RBLUP is not suitable for polyploids, I think.
Okay, So yeah, so probably on a stage wise, we'll be able to do that because Jeff Hindeman was the first that published these results with those effects.
And the answer is implicit in the question. Like if you are using dosage for your genomic relationship matrix, so it will be taken into account if you are using the ploidy.
Sanjeev, I think I couldn't get what it said. It is.
If
I cannot understand.
When you are making your genomic relationship matrix for genomic selection using dosage data, so that additive effect will be taken into account in the way relationship matrix will be constructed.
So I'm not sure if I got it correctly, but you can translate from one to the other. So they are quite the same. When you run an R-BLUP and a G-BLUP using dosages, you can get the dosage effect from the G-BLUP as well if you want to, if you have the dosage, right?
Chris?
Yes.
On line 27, if you try changing that ploidy from 4 to 3 and run that, what happens?
I don't know. Let's see.
It should run, right? Okay, it runs. Okay, yeah, that's exactly what I was getting, and I don't know. Or maybe it's a warning, not an error necessarily, but
it is an error. I don't know if they allowed.
But in theory, it should work, right? I mean, here I'm saying, give me.
I don't know if it's only odd ploidies.
Okay, that could be one of the reasons.
Yes, maybe if you put 6 instead.
It looks like, yeah, it has to be an odd or even number of ploidy rather than odd.
Try 5, because if you do 3 and you had 4s, then it might have an error.
Okay, now I let it run or I change it.
But you might have to wait so it doesn't crash.
Yeah. And there is a question about installation of Docker in Apple. I never tried, but I can help you. Maybe after this session ends, I can stay here with you and help you with that. Okay. And I don't know, I think the session's supposed to be only 1 hour, no? And
I think it's supposed to go into 2, but
okay.
I'm sure we can keep going.
Yes, we can keep
and
going.
I think the Apple Silicon Macs have native Docker now. So I think everything will translate over. And if it doesn't translate over, it should, in theory, it should then run on Rosetta and it should translate it for you. But I'm not 100% sure. But Chris and I can stay on after and see if it can get it to work.
If we have more time, I don't know if I miss any questions in the chat. Sorry if I didn't answer your question. Please, if you can send it again. I kind of get lost in the list. But if we have time, I can go through the tutorial too and show how it worked with 6. And probably now we have, and I can show 5 doesn't work too. I don't know, maybe it's even number issue. Okay, should I go to the tutorial? Someone wants to ask any other questions? Or okay. Okay, start from the beginning. And the idea is that for this tutorial, you don't need to have much resources, but you do need to have Docker or Singularity. And I will use these images. So I already have this in my computer. This will take a little time to load. And once you load, if you are using like a desktop version of Docker, you can see the containers here and your image, like which ones are used, which ones not, which the version. And these are all the images that I have in my computer. And sometimes, warning, sometimes it can occupy a little space in your computer. So if you don't want to use them anymore, and just come and delete here and delete the container and just to free space in your computer. And next time that you use it, just load again. And so I have this, and I have computer here that I can use for that. I would just put that here. And the example dataset is a rows dataset, so I have just 4 samples, and just to be easy to run. So I will create a directory here called tools 2023, and I will download all the data files, the FASTQ files, and the FASTA file, that's the genome that I will use. And just download all of it. And for who never saw a FASTA file before, it looks like this, and has a first line with a reader, a header, and the second line with the sequence by itself, and a line with the quality measurements. And these letters will indicate a measurement, a quality measurement. It depends of your sequence or sequencer, and you have 4 lines by sequence. And so this is the FASTQ file, and I also have the genome file. That's the chromosome for FASTA, and it's just a single chromosome because I didn't want it to occupy too much space. It's here, all the TCG. And first I would do this step by step, and then I will do this with the workflows, and you will see the difference. This is just a subset. I will not require too much computational resources, but I put this graph here just to show, just so you to have idea of how much RAM and how much CPUs and all the stuff that you need to run an entire data set. This was how much I use it for the rows data set. So first, for the quality control, there's a very famous software that's the FASTQ, and it provides like very nice reports about each one of the FASTQ files, and it's running. So what I did was, because I'm running my computer, I'm running with Docker. So I just asked Docker to run, and this -v means that I'm taking my working directory, like the current one, and putting inside the container as the OPT, and then I just put the name of the container that I'm using and the command that I want to use, and indicate inside the container where are my files and how I want to use it. And so this is all the FASTQ GZ. I just did a loop because I don't want to put like 4 lines here, so I just did a for loop in all the FASTQ files. And this is with Docker. If I wanted to use Singularity, it's the same logic, but instead of Docker, use Singularity, exec, and instead of -v, you put bind here. And instead of just putting the Docker container address, you point to an image, SIF image that you loaded up here. And you just indicate the path to this image that you loaded. And the result of it, so the results will be HTML report. The ZIP contain all the images, and if you, I put an example here of this HTML. So the FASTQ outputs by sample, and they provide many statistics like the quality per base based on that 30 line of the FASTQ file. Many of them, but it's important to see that this software was made for whole genome sequence. And then sometimes, like for example, the GC content and the per base content, it's messed up because it's GBS data. So of course, this is not as random as supposed to, because we have a restriction enzyme here. And this also, we have PCR duplicates, so this would be a mess. So it's not because it's red here that's wrong, it's just because it's GBS data. But I don't, like usually I don't have only one sample, I have many samples. In this population I had 122. And So the multi-KC software kind of do an overview of all the reports. So I'll come here and just run Docker again. I'm using this image, and I will generate the tutorial samples report. So instead of seeing just one sample, oops. It makes like a summary of all the samples. So the number of duplicates, because it's GPS, this is high. And number of sequences, this is also interactive, and you can export. I really like this report, the multi-KC reports, the quality and by sample. This data set, I already cleaned it, so it's always expected. Like these GC contents because of the PCR duplicates, and I already removed the adapters. So this sequence, it's ready to go, but I included in the tutorial some software that you can use if you find issues, like if you find adapters, if the quality of your sequence is not good, and you want to clean. And I provided some software there that you can use. So R is good. So I will go ahead and do the alignment. And because I'm using GATK suggests to use BWMN to do the alignment. So it's what I will use. And I have other images here for that. And before doing the alignment, I need to do the index files for the genome, for the reference genome. And I will do this with the BWA index and with the Picard. And I will just come here and run this. And then we run fast because my reference genome is small. And I will have now my genome that was already here, but all other auxiliary files that's required for run the BWA. And I will not run one by one. I will do a loop, and then I did a loop in the FASTKey files, and I will run the BWA, and I will define the samples and the alignment files here. And also I will do the sorting and indexing the PAN files in the same loop. In here, and do that. Oops, sorry. Some of these commands, and I know that BWA have this, like has this option here, that's the threads that I'm using. And this will be useful if you want to parallelize. Like I'm running this in my computer, so I will not parallelize in many cores, but if you're using a high performance computer, this can be increased and it make faster your analysis. And I, in the tutorial, include the Docker and also the singularity options for each one of the commands. In the next step, I will do another index for the reference genome, and then I will run the 3 steps of GATK. One is the haplotype caller that will call the genotypes by individual. So you have a VCF file, a partial VCF file for each one of your samples. And then you will include this in a database. That's the second part, because in GATK, they work more with humans, and then they don't have like an entire population ready. So, and you run the SNP calling this population, but they have individuals that ask to do like 23andMe or things like that. So samples come one by one, and the only thing that they do is include the sample inside this database. So, and it's a good thing because doing by sample, we can also parallelize this procedure easily. In the workflow, what I do, I split the number of samples in batches. So imagine that I have like 200 individuals. I will do in batches of 20 or batches of 4. And each one of the nodes of the computer, I'll use 40 cores. And then each one of the batches will run in one node. So it makes easy also for parallelize and optimize the computational resources. And I think the alignment's done. So now I also have the BAM files, the alignment files, and the index for it. And now I use that as input for the GATK. Oops. So this only creates the FASTA file. And this will take some time. Oops. Here. Take some time to run.
Sorry, I just have a question.
Yes.
When you're using like the Docker or Singularity, do you have sudo access to run them on the HPC?
When I use Docker, I am in my computer, so I have sudo permissions, but I included the Singularity here because Singularity, you don't need it. So usually HPRC or HPC already have Singularity installed, and you just need to download the image as SIF, and every time that you use the image, you go to these files. Like, see here, the Singularity option. You go, instead of pointing the image to download it from internet, you just point the image that you downloaded as a SIF. And this don't need sudo permission. You can do that, the HPC.
Okay, thank you.
I think it has some issue, probably. Oops. Okay. Now it's going. Any other questions? Let me see if I can tell about other things while it's running. Maybe I can already set up the workflows to work. So what I did here, I did step by step on how to do it with GATK. And I'm not doing some steps that's also suggested by GATK. That's a hard filtering after that. And I think I included the description of how would be the hard filtering here, what's that's for. And GATK, it's amazing about tutorials and documentation for all their tools. And sometimes even too much, like it's an entire word and you need to spend some time learning that. And this is one of them. But luckily, I did that already and during my PhD, and then I include this inside the workflows. So with the workflows, you can have like this overview of everything that's happened inside. So this is the main workflow, and I will create the alignments here in the first sub workflow, and then I will run Freebase and run GATK. So inside this GATK is the same thing that we were doing step by step, but I included other things like the hard filtering, also a normalization of the indels position with PCF tools, and I included other options here. And the alignment sub workflow will do the same thing that we did, but we will split this in nodes and cores. And the final task will merge all the bands because some softwares required this to be merged. So I already included that. And the Freebase, and then I run Freebase too. and in the workflow, and I split the genome positions by cars and nodes. So it's optimized, and I also did the normalization of the indels positions using BCF tools. And then someone asked about the links, all the links in the chats. I can ask Delaney to put the links in the website, the Tools for Polyploids website. So it will be there. And the presentation too. It's recorded and she will include this in the website too. Let me see. Okay, finished. So now I have the GVCF files. This is already a VCF, but it's just for a single sample. So as I showed before, like that scary heater, and then the genotypes for a single sample. And then the next step here, I will create a database. Or first, I will create a list of intervals and just some bash commands here. But at the end, what I want is a list of this interval list. That's just a list of chromosomes. Here, just have one, but we usually have like chromosomes and scaffolds if the genome's like a draft or something like that. And so I did the commands for doing this automatically. And once I have this, I can run to get the database. Oops, not Singularity. It's the Docker. Well, here. Here. And then finally get a combined VCF. Okay, and this is the final VCF. Has all the header, and then the genotype information for my 4 samples. And I already explained to you what it means. That's this is missing data. I don't know if I mentioned before. Okay. Okay, this is how we do step by step. And now I will show you how to do with the workflows. So I will need the workflow by itself, and my page, like you can find other workflows to work in other things in the repositories. You remember like Docstar and Workflow Hub. So if you don't want to do these specific things, you can search for other ones there. And the workflows that I'm working in, it's already available. And I usually, when it's okay to be used, I make releases. So I did this recently, like this specific one. And what you need to do is download these two files. And this is the main workflow, and the zip file is all dependencies because I use sub workflows and tasks and everything. So it's just these 2 files. So go back here. I put these links here just to make it easy. Download everything. And I need to create an input file. That in this input file, I will define what I want inside to run or not in the workflows. So this is the maximum cars that I will use. This is deployed. If I want to remove the duplicates or not, and because it's a GBS data, I would not. If I will run GATK, if I will run Freebase, if I will run the hard filterings, how many chromosomes I have, and this is for parallelization also. And this is the for parallelization also. Like if I would distribute the samples in nodes, how many samples I will have in each one of the nodes. And this is the samples file. I just need to describe my samples. And it's 3 columns, one with the file name and 2 others with the sample name. And if you have like duplicates of same sample, you should define the library in the last one. So GATK can consider like the variation between duplicates. So maybe it's a good idea to keep them separate. So the first one's sample, and then the third one is unique spots in your experiment. So this is the file, and then I need to give also all the reference genome and the index of the reference that we already generated up in the tutorial. So probably I finished to download everything that I needed. So the Cromwell is here. Okay. And then I need to do this file. Use Max for that. Okay. Here, I just save it. And then I will create a sample file. This file. That's this here. Save it. So now I have the empirical here. The sample file here. Good. And if you will run this in HPC systems, you need to have a specific configuration file to the workflow connect and talk to your management system. So I included in the repository configurations for Slurm and SGE. I confess that I never tested in SGE systems, so if you have issue, let me know. But it works here with the Slurm system. And you just need to change the default backend configuration in this file here. And you need to include this in the line to run the workflow. I'm running in my personal computer, so I use Docker instead, and I don't need a specific configuration. So just run this one. Oops. Oh well, here. So, okay. I didn't. Okay, it's running. So it will output many things, and it's a log of all the steps that they are doing. So it's running the first. I'm pointing. I should use my mouse, but it's running the first task. Oh, it couldn't find the samples in. Let me see. That fails first. But so what I'm doing here is running a command that I put the main workflow, that's the WL workflow, the input file that I just included here, the zip file that has all the dependencies. So it's just this line. And it fails because couldn't find the sample file. Samples file is here.
You have it miswritten as samples info, and you have it, the file name is samples file.
Oh, okay. Oh, thank you. Here. Sample files. It's like this, correct? No?
Nope. You have it in the script. You have it written as samples _ info.
Oh, _ info, like this.
Yep.
Okay. So here it's just. Oh, great. Thank you. Now should run.
Famous last words, Chris.
Okay. So it will submit all tasks and you just watch. And that's it. And you hope it doesn't break. Oh, something that's really good. It's that you can also include a cache. You can connect the workflow with a MySQL database. So if happens that the workflow crash, and sometimes it does, because mostly if you are working with HPC, if you don't allocate enough memory or CPU, it will break because you didn't. So you need to adjust the parameters for the runtime, and it can happen. And if it breaks and you have a cache, you don't need to run all the tasks that you ran it before. The workflow runs just the task that broke. And if you have like a weak internet connection, this also can happen, and so on. So it's good to have a MySQL configured, and it's can be a little, depend of your HPC, it can be a little hard to configure, but people from the TI maybe can help you. But once it's done, works good. I use WDL, like Widow language to do that, because I was already working with G80K, but I know that's possible also to convert the scripts to CWL that has other types of ways of running that maybe is easier to configure cache and in HPC. But I never tried, but I know it's possible. I have a question in the chat asking for those who doesn't have HPC available, how they can use it. So if you don't have HPC available, the good things of the workflows is that you can use cloud system too, that it's available for everybody. So, and in the Terra platform, it's specific for WDL workflows. So you have like a graphical interface that you can like get my workflow from the docstar repository and submit your samples and everything on a cloud environment, in the Google Cloud environment, and you can have access to the resources like this. And of course, you need to pay for the amount of like the computational resource that you use, but it's possible and it's easy actually. For this type of language, it's easier to run in cloud than running HPC, because HPC you will need to do all this configuration stuff, and in cloud this is already made for you. So it's a great advantage of using this type of systems too. And so you can the Docker and the Singularity images will make that you don't need to install things. So if you open a machine on cloud, you don't need to install all the softwares. You just call the image. And if you use like the Terra or even Galaxy, or if you have like, if you want to use a Galaxy workflow, you can do this on your browser in the web platforms that they provide. So you just point the image that you want to use and the workflows that you want to use, and it's done. And I participate of a course of Broad Institute in Broad Institute in a conference that I went, and I was planning to try to do like a workspace in cloud for reads to map to in Terra. But I still didn't have time to do that, but it's in my plans. So to make it easy to run this in cloud too, in cloud environments, and it's I will be there. But if not, it's already possible. But you need to study a little bit how the platform works. So it's done, and then you have the outputs here, and of course I included more things in the workflow, so you have more results. Like I included some output of the quality graphics that you can access in this file, like this hard filtering graphics here, like you see. Sorry, I will not explain, but outputted these graphics in the workflow too. I outputted the merged then if you want to use the alignment files for other things, and like other VCF evaluations, and for the freebase VCF and for the JTK VCF, and here are the VCFs. So this is the JTK one. I can just show you really quick the VCFs here. So it's the same thing, and that's it. The VCF for the JTK, and the VCF for the freebase. It's also here, listed here. You can see here. And it's the same things, but instead of using JTK's freebase command here. And you also have, no, you don't have. I don't know what happened. Sorry, guys. Freebase didn't work very well. I will fix this issue later. But, or I don't know if it's few samples too. Sometimes, yes, probably. But that's it. I will check what happened here later. Maybe it's few samples, but it's working in the complete data set. I'm sure of it because I'm running other data set. That's it. If you have more questions, we will be here. Let me see if I forgot something. No, and then once I have these VCFs, it doesn't matter how you got these VCFs, or if you have HapMap, or maybe if you have like the DartSeq also, you have these counts of each allele, and you can go through the dosage column that I already showed you earlier, how to do these steps here. Okay, I included tutorials links everywhere, but if I forgot some, just let me know and I can share with you. I'm in Slack, and I also put my mail here if you want more links. It's here.
I'll just put the Slack invite link if you want to join. You can find both me and Chris there. You'll be able to find, I think Gabriel's also in there. Maria's going to be in there, so you can reach out to us.
I will put again the links of these materials in the chat. It's already available. Here. Thanks everybody for being here listening until now, and for the questions too. It's very nice.